Mivarts Dayama and Preadaptation. St. Geoffrey Jackson Mivart, 1871, wrote in his book, On the Genesis of Species, that he could not see how evolution would proceed by the gradual accumulation of small changes, since most intermediate character states would be maladaptive. For example, what use is a partially formed winged torreptile? He, therefore, suggested that evolution would proceed in jumps, and that basis of these was the acquisition of useful characters. Mivart's dilemma has been explained by invoking preadaptation. The intermediate structures act in a peculiar way for which they are well suited, but by chance they are appropriate for other roles which they are able to play after further elaboration. For example, a flap of skin between the forelimb and the trunk might have evolved in reptiles as a thermoregulatory device and then have been found of use in gliding away from enemies, ultimately giving rise to a wing. Similarly, the air bladder of fishes was not evolved so that they might one day invade land but as an accessory respiratory organ for use in the aquatic environment. However, once evolved, air bladders were of use as lungs when new environmental problems, such as the drying of ponds, appeared. See Callow, 1983. Microevolution, macroevolution, megaevolution, and hypothesis of punctuated equilibria. Goldschmidt 1940 divided evolution into microevolution, that of subspecies, and macroevolution, that of species and genera and perhaps also of higher categories. Simpson 1953 has proposed the additional term, megaevolution, for really large-scale evolution, such as that of families, orders, classes and phyla. Evolution at these levels receives much attention from students of the fossil record. B. Wrench 1959 has modified the terms microevolution into infraspecific evolution and macroevolution into transspecific evolution. Callow 1983 has regarded macroevolution and cladogenesis as synonyms. For several years, 1976 to 1982, Stephen Jay Gould and Niles Eldredge of the American Museum of Natural History have questioned the conventional view that evolutionary changes in the distant past are principally the outcome of the gradual accumulation of slight inherited variations. They advocated that most evolutionary changes have consisted of rapid bursts of speciation alternating with long periods in which the individual species remain virtually unmodified. Gould and Eldredge maintain that most lineages display such limited morphological changes for long intervals of geologic time as to remain in stasis, or in equilibria. Conspicuous or prominent evolutionary changes are concentrated in those brief periods, punctuations, when the lineages actually split or branch. This is called hypothesis of punctuated equilibria. Further, branching from a single lineage, or a single line of descent, during macroevolution, will ultimately produce a cluster of lineages known as clade. Thus, branching sometimes is called cladogenesis. When referring to a clade or to a group of related clades, it is common to use the adjective 104 evolution, evolutionary biology. Phylogenetic, not to be confused with the similar word phyletic. Phylogenetic pertains to a portion of phylogeny consisting of more than one lineage, whereas phyletic refers to a single lineage. Moreover, a lineage reconstructed from fossil data may exhibit sufficient evolutionary change that a taxonomist DMC appropriate totavididento tointergrading species. Such species are known as chronospecies, successional species, paleospecies or evolutionary species. See Stanley, 1979. 1. Microevolution. Evolutionary changes in populations are ordinarily visualized as gradual, built upon many small genetic variations that arise and are passed on from generation to generation. The shifting gene frequencies in local populations may be thought of as microevolution. The progressive replacement of light-colored mobs by dark, melanic mobs in industrial regions in England, i.e., industrial melanism, exemplified SE microevolution. Most population geneticists endorse the view that the same microevolutionary processes have been involved in the major transformations of organisms over longer span of geologic time, macroevolution. The traditional outlook is that small variations gradually accumulate in evolving lineages over periods of millions of years. 2. Macroevolution. The most significant feature of macroevolution is a progressive, sustained tendency for certain characters to develop along an evolutionary line. Trends, or directions of evolution, of this sort are numerous in the fossil record. Long-term progressing trends rarely appear in only one structure, but almost always involve a complex of different features. In fact, trends are produced by the driving force of natural selection operating within the limits of a particular adaptive zone or subzone. Evolution is not random, although certain elements in the process are random, and trends leading to greater efficiency are to be expected. Evolutionary trends are generally adaptive movements along one pathway, but they are never exclusively sequential and always involve divergent and repeated taking on of one or the other characters important in the trend. The populations undergoing change are constantly experimenting within the adaptive pathway, and parallel probings of a new subzone by related but different lines are the rule. The classical example of evolutionary trends in macroevolution is provided by the horse family, Equidae. Evolution of horses. In reconstructing the phylogeny of an animal group, it has been the standard practice to show the emergence from a central, generalized stock of a large number of divergent branches or lineages. Nodal branches persist, indeed the general rule is that all but few perish. The disappearance of many branches in the distant past might lead the observer today to the mistaken impression that the evolution of a particular group was not at all elaborately forked. Thus, the evolution of horses could be erroneously depicted as an undeviating straight line progression from the small, terrier, small dog of various breeds, sized Hyracotherium, Eohippus, to the large modern horse, Equus. On the contrary, a wealth of evidence has revealed convincingly a pattern of many divergent lineages, figure 8.12. Figure 8.12 demonstrates very nicely the gradual change from a dog-like, browsing creature with padded feet to a horse with grazing habits and hoof-springing feet. In fact, the appearance of the anedode condition was a landmark in horse history. The fleet-footed, one-toed Pleohippus emerged during the Pliocene epoch from the slow-footed, three-toed Merychippus. This major event is usually interpreted as a gradual change within a single lineage. In other words, a direct ancestral descendant relationship between Merychippus and Pleohippus has been widely accepted. The paleontologist Simpson, 1951, departed from the view of slow and steady change when he suggested that such a major transition might have involved the accumulation of small genetic changes in relatively rapid succession. This accelerated pace of phyletic gradualism was called quantum evolution by Simpson. Proponents of hypothesis of punctuated equilibria explained the sudden appearance of Pleohippus in a quite contrast but convincing way. Thus, one might argue that Merychippus existed for a long period with little or no evolutionary change, stasis, and eventually suffered extinction. Pleohippus is not a linear descendant of Merychippus, but a dramatically new form that had earlier arisen as a small evolution above species level 105. Peripheral population of the parent species. It may be noticed, figure 8.12, that not all offshoots of Merychippus evolved the progressive single-toed condition. Several lines of Pliocene horses, such as Hipparion and Nanippus, retained the conservative three-toed pattern. These conservative Pliocene horses were evolutionary blind alleys, and may well have represented punctuations that failed. Recnt. Pleis to cene. Pilocene. Miocene. South America, North America, Old World. U. 2.2. Equus equals toes 1 to 1. Dot equals springing equals lst wilo bg b Hipparion equals I e u Piahippus o nanup u sneo underscore tri equals less than Hipparion equals 106 EVOLUTION Evolutionary Biology. General principles of macroevolution. The following common features have been derived from the phylogeny of horses regarding macroevolution. 1. Macroevolution follows the acquisition of new general adaptation or entrance into a new adaptive zone. For example, Darwin's finches radiated after an apparently generalized finch ancestor arrived to occupy the previously unoccupied Galapagos Islands. Reptiles radiated after completely terrestrial development was established as a general adaptation. 2. Macroevolution always involves evolutionary divergence. Macroevolution is not linear but radiating. Usually radiation follows general adaptation and the invasion of a new adaptive zone through special adaptation in different divergent descendant lines. 3. Adaptive radiation or macroevolution tends to produce evolutionary lines that converge in special adaptation with other distantly related groups differing in their matrix of general adaptation. For example, ichthyosaurs show a marked evolutionary convergence with a number of fish groups in their body form, manner of locomotion, food habits, fishes, and free-swimming pelagic life. They are convergent in special adaptations but still share a group of general adaptations with all other reptiles. 4. Macroevolution produces groups of parallel special adaptations among divergent but related stocks. All of them have a common general adaptation. Among reptiles, group after group representatives of every order except the Cotylosaurs had invaded aquatic adaptive zones and become completely aquatic. Among these, some groups flourish and later become extinct. These groups were replaced by other parallel groups which invaded the same zone and underwent differentiation. The Phytosaurs Triassic, of the order Thecodontia, and the crocodiles Triassic to recent, of the order Crocodilia, invaded the aquatic adaptive zone. But later phytosaurs became extinct and then the crocodiles replaced them. These provide an example of parallelism and ecologic replacement. Thus, where there are repeated evolutionary experiments with the same broad group of zones, some groups arise, flourish and become extinct to be replaced by parallel groups that invade the same zone and undergo differentiation in their turn. 5. Macroevolution ultimately leads to extinction. Some groups acquired special adaptations for a narrow adaptive subzones and due to these specialized adaptations they could not move into new major zones. Because all adaptive zones finally change and disappear, so all groups restricted to a narrow zone also disappeared. Hence, the groups which could change the adaptive zones by acquiring general adaptation survived and others became extinct. 3. Megaevolution. Megaevolution, or the origin of new biological organizational plans, is rare and shares features common to microevolution and macroevolution. Only a few major general types have developed during the history of life, but almost all of them persist without extinction, although a few have perished and others are relict. All of the phyla and most classes of microorganisms, plants and animals represent marvelous complex and coordinated groups of general adaptations, each forming a basic, distinctive biological organization, unique and dominant in a broad range of characteristic adaptive zones. About 200 of these major biological organization plans are known to have developed in last 3 billion years. The origin of these systems is the most significant of all evolutionary events, yet the processes leading to these events remain the least studied of biological problems. Megaevolution has the following characteristics. 1. The breakthrough always follows evolutionary experimentation and exploration by divergent lines of the ancestral stock, until one of them crosses the ecologic barrier into the new zones. 2. The breakthrough and shift are always rapid, otherwise they fail because of the extreme negative selection in unstable ecologic zones. 3. The new zone is always ecologically accessible, is devoid of competition and requires a new general adaptive type for its invasion. 4. Adaptive radiation always follows the initial shift. Evolution above species level 107. The process involved in macroevolution and megaevolution. The major feature of organic evolution is divergence guided by the molding force of natural selection. Evolution at the populational level is driven by the elemental forces of mutation, selection and drift. Basically, population evolution may be sequential, but it is always divergent in the end. Speciation, macroevolution and megaevolution represent states or levels in a continuum of evolution. All are driven by the elemental forces but are subject to increasingly complicated effects from less understood forces as well. Selection becomes of greater significance above the microevolutionary level. Of the greatest importance in speciation is isolation. Selection may act to produce divergence in this process whenever fragments of an originally interbreeding gene pool become spatially or reproductively isolated. New species originated from old ones through the origin of reproductive isolation and independent microevolution. Thus, in addition to elementary forces, isolation, microevolution and macroevolution, megaevolution involves the following processes. Figure 8.13. 1. The possession of new general adaptation or occupancy of a new adaptive zone. 2. The breakthrough into new zones or subzones within the new adaptive zone by development of special adaptation. 3. The loss of evolutionary flexibility, and channelization into greater and greater specialization within subzones. 4. The ecological reinvasion of a zone or subzone when it becomes partially unoccupied because its original occupiers are now specially adapted, ecologic replacement. 5. The irreversibility of evolution. Since each step is dependent upon the previous progressive changes, once a group is on an adaptive erode, it is usually trapped in an adaptive zone and cannot traverse evolution against the direction of selection. 4. Doctrine of punctuated equilibria. A single line of descent or lineage may persist for long reaches of geologic time. As small changes accumulate over periods of millions of years within one lineage, the descendant populations may eventually be recognized as a species distinct from the antecedent population. The persistent interbreeding blurs the distinction between species. A. The myrtle warbler and B. Audubon's warbler were formerly thought to be two separate species but are now considered to be merely local varieties of one widespread species. Accumulation of small changes within a lineage has been termed phyletic gradualism, and the transformation of a lineage over time has been termed as anagenesis. How organisms come to terms with and keep in tune with their environment is called anagenesis. See Callow, 1983. As depicted in Figure 8.14a, a new species, labeled B, arises from the slow and steady transformation of a large antecedent population A. If only transformation occurred, then life would cease as single lineages became extinct. Hence, a new species, C, can also arise by the splitting or branching of a lineage. As 108 evolution, evolutionary biology. Already described, the extinction, splitting of one phyletic irreversibility. Lineage into two or more diversification. Linesis termed cladogenesis, viz., the evolution environment. The prominent episodes of evolution in the history of life are associated with the splitting of lineages, but the splitting is not seen as slow and steady cladogenesis. The new species arises through rapid evolution when a small local population becomes isolated at the margin of the geographic range of the parent species, Figure 8.14b. Indeed, the successful branching of a small isolated population from the periphery of the parental range virtually assures the rapid origin of a new species. In geologic time, the branching is sudden, thousands of years or less, compared to the millions of years of longevity of the species itself. Figure 8.13. The process of evolution above the species level, a diagrammatic representation of macroevolution and megaevolution, after Savage. 1969. The punctuational change does not cause any unconventional evolutionary phenomenon. The sudden appearance of a species is fully consistent with Ernst Mayr's rapid selection in peripheral isolates. The morphological differentiation of geographical isolates occurs early and rapidly, especially when the population is small and adapting to the new abnormal environment at the periphery of the range. Once established as a new species, the assumption is that little evolutionary change will occur in that species over geologic time. A well-studied example of punctuational mode of evolution is provided by Peter G. Williams in 1981. He studied the fossilized freshwater snails, mollusks, in the Lake Turkana's basin region of Africa, northern Kenya. The thick, undisturbed fossil beds contain millions of preserved shells representing at least 19 species of snails. Several lineages of Cenozoic snails were exceptionally stable for 3 to 5 million years. When morphological changes in shell shape did occur, they were concentrated in brief periods of 5,000, 50,000 years. These rapidly evolved new populations then persisted virtually unmodified until they became extinct. Some authors have convincingly commented that so-called sudden changes within 50,000 years might appear to be instantaneous to a paleontologist but would be almost an infinity to an experimental geneticist. In fact, to many geneticists, an interval of 50,000 years, or 20 generations in snails, would be sufficient time for the morphological changes in the snail populations to accumulate gradually rather than dramatically. Evolution above species level 109. Species C, species B, species A, species B. Trademark. Figure 8.14. According to phyletic gradualism, species A gradually transforms into species B, and may split into as slowly evolving species C. B. According to theory of punctuated equilibria, a small isolate from the parental population, species A evolves rapidly into a new entity, species B. Species A undergoes a long period of stasis during which little or no morphological change takes place. Species B does undergo minor structural modification with time, after Volpe, 1985. Whether human evolution is gradual or punctuated. On the basis of phyletic gradualism mode of evolution, it has been standard practice to put Homo sapiens at the end of a continuous series tracing back through at least two other species, figure 8.15. According to certain modern authors such as Stanley 1979, Callow 1983, and Volpe 1985, various taxa involved in human phylogeny are the following. 1. Ramapithecus. This group also includes Subapithecus. These ancestral forms, apes, lived approximately 8-14 million years ago and contained a flattened face with a short muzzle. First fossil, Afragileoperja, of Ramapithecus was recovered by G. Edward Lewis, 1930, in Shivalik Hills of northern India. Another fossil of this group was recovered from Lake Victoria in Africa by Leakey in 1955. The fossil evidence clearly indicated that such genera as Ramapithecus, from Rama equals Hindu god plus Pithecos equals Greek for ape, and Subapithecus, from Shiva equals Hindu god, which lived in Africa and Eurasia, were the forerunners of hominids. 2. Australopithecus afarensis. L. Austral equals southern. The fossil of this female ape man, called Lucy, was recovered from a far locality in Hadar, Ethiopia by Donald Johanson, 1973. This fossil was probably the oldest among all the primitive man described. The afarensis remains dated between 3 and 4 million years, ago. 3. Australopithecus. This group lived 1.5 ago. They had a flattened face. The orientation of the skull relative to the spinal cord, and the bones of the hind limbs relative. The orientation of the skull relative to the spinal cord, and the bones of the hind limbs relative to the pelvis suggest an upright gait. There are following two groups of these ape men. I. Robust Australopithecines, Australopithecus robustus, which had a heavy skull and dentition suggesting a vegetation diet. The fossil remains of this group was recovered by Robert Broom and Raymond A. Dart, 1938, from caves in Sterkfontein, Kromdraai and Swartkrans in South Africa. E. Gracile Australopithecines, Australopithecus africanus, which has a more delicate skeleton, jaw was light and slender, and supplemented their diet with animal food, i.e., they had smaller molars than the A. robustus. First fossil remains of A. africanus were obtained by R. A. Dart in 1924 from a 110 EVOLUTION, Evolutionary Biology. Limestone quarry near Tong village in Transvaal region of South Africa. Lewis Leakey and his wife Mara Leakey, 1959, uncovered bony fragments of a robust Australopithecine with enormous teeth from Olduvai Gorge in Tanzania. It was called Nutcracker Man or Zinjanthropus and was about 1.8 million years old. Zinjanthropus is considered an East African variety of A. robustus and has been assigned a status of separate species, Australopithecus boisei. 4. Homo habilis, handy man. The fossil remains of fish hominid were recovered by Leakey in 1964 from rocks of Olduvai Gorge in East Africa. This group lived about 1.5-2 million years ago. They had an erect gait, relatively large brains and were tool users. They fabricated crude tools out of pebbles and used them to gather a variety of plant materials, nuts, seeds, fruits, tubers and roots, and small animals, lizards and rodents. Cooperative behavior evolved in them and promoted the sharing of the food for the first time. 5. Homo erectus, upright walking man. This group lived 1 million years ago. They had a gait of more like ours and larger brains than the habilines. H. erectus used tools of bone and stone and used fire. Fossil remains of H. erectus are discovered at Trinil, Java, in 1894 by Eugene Dubois. They have been called Javaman and had a cranial capacity of 775 to 1000 cubic centimeters, or cc. Another type of fossils of H. erectus, called Peking fossils, i.e., Peking men, were discovered by Davidson Black in 1920s. Peking men have the cranial capacity of 900 to 1200 cc. 6. Homo sapiens, thinking man. The grade of sapiens contains at least two contrastingly different anatomical types, the bulkily built and heavily muscled Neanderthal and slim-bodied Cro-Magnons. The skeleton of Neanderthal man, Homo sapiens neanderthalensis, was first unearthed in 1856 in a limestone cave in the Neander ravine near Dusseldorf, Germany. The Neanderthals were 1.5 lakh years old cave dwellers, short, about 5 feet, but powerfully built, with prominent facial brow ridges. They had large brains with an aver. Homo sapiens. Milleons. Ofyears. Ago. Figure 8. Australopithecus afarensis followed by Homo habilis and Homo erectus are believed to have existed successively in geological time and to form a lineage leading to Homo sapiens. In the distant past, the Homo assemblage coexisted with the Australopithecus groups. This phylogeny is based on the views by Donald Johanson, 1979, after Volpe, 1985. A8. A fossil skull of Homo erectus show that the human brain gradually grew larger, beginning about 2.5 million years ago. At the same time the ridges above the eyes became smaller, and the face and jaw began to jut out less. Increasing intelligence and the ability to make better tools apparently went together. B. The earliest stone tools, made in Africa about 2.5 million years ago, were simpler than the hand axe shown here. Evolution above species level 111. Age capacity of 1,450 cc as opposed to 1,350 cc in modern humans. Neanderthals made complex stone tools, were accomplished hunters of large game, used animal hides as clothing and withstood the rigors of the bitter cold climate of the last glaciation. There is evidence that the Neanderthals buried their dead with various ritual objects. The fossil remains of Cro-Magnons, Homo sapiens sapiens, a representative of our species, Homo sapiens, were unearthed from a French rock shelter in the village of Les Esquis. They were about 35,000 years old. The Cro-Magnon people occurred in Europe and left behind very elaborate cave paintings showing attainment of a form of culture not unlike our own. We do not know about the birthplace of modern Homo sapiens. Modern humans may have been cradled in Asia or Africa. The skull of Neanderthals suggests a brain at. In the past 3 decades, Africa has become in Euro-Astas largest the top modern humans. They were very strong and stocky and seem to increasingly the focus for investigations into human origins, being well suited to the Ice Age climate and differentiation. Several sites in Africa have yielded fossils that reveal different phases of the human story from the earliest stages. The differences of opinion that exist concerning the phylogeny of the hominidae are vividly revealed by the ever-changing reconstructions of the human and evolutionary tree, almost on a yearly basis. One of the most widely held views is that a gradual ancestral descendant relationship existed for each successive hominid group. Thus, the Australopithecines shaded faintly into Homo habilis, who in turn graded slowly into Homo sapiens. The theory of punctuated equilibria claims, C. Stanley, 1979, that the evolutionary changes in hominids occurred in rapid bursts, concentrated in speciation events, figure 8.16. Such Homo sapiens, Telomo erectus, TT Homo habilis, O. Australopithecus robustus smiley face, O0, I, 2, BC, smiley face, W. Punctuated episodes were separated by long periods of stasis in hominid lineage during which little or no morphological change took place. Such long periods of stasis occurred within both the Australopithecine and hominid groups, and this is rather startling considering the high net rate of evolution which is characteristic of human evolution. Figure 8.16 shows that this pattern of evolution appears rectangular, rather than tree-like. 112 EVOLUTION, Evolutionary Biology. Simpson's Ho PEFUL Mo NS Ter. There are genetic changes, known as homeotic mutations, which are responsible for such dramatic developmental effects as the transformation of one major organ into another. In the fruit fly, homeotic mutations transform an antenna into a leg, or a balancer into a wing, or an eye into an antenna. Although fascinating for developmental studies, most evolutionists would not even consider that the origin of a new species could ever involve such drastic major morphological changes produced by single homeotic gene changes or macromutation. However, in 1940s, the prominent geneticist Richard Goldschmidt asserted that homeotic mutants do illustrate a possible mode of origin of novel types of body structure, and, hence, could be responsible for the rapid emergence of a new species in the distant past. He acknowledged that the vast majority of macromutation would have disastrous consequences, monsters. An occasional macromutation, however, might conceivably immediately adapt the organism to a new way of life, a hopeful monster, in Goldschmidt's terminology. Goldschmidt was convinced that the gradual accumulation of small mutations was too slow a process to account for broad macroevolutionary trends. He favored the idea of a dramatic, abrupt transformation of a population by a favorable, instantaneous macromutation, which could take the form of a radical rearrangement of genetic material, viz., a gross chromosomal change. The concept of punctuated equilibria does not require or imply chance-favorable macromutations. Rather, in the process of allopatric speciation in peripheral isolates, the incipient species develop their distinctive features rapidly. However, the significance of one aspect of Goldschmidt's views cannot be denied. Structural chromosomal rearrangements may play a primary role in the initiation or achievement of reproductive isolation between two diverging populations. Orthogenesis and orthoselectation. The observed tendency of a part or organ to change progressively in size is called orthogenesis or evolution in a straight line, Moody, 1970. The theory of orthogenesis, which was originally proposed by Eimer, 1897, lays stress upon the fact shown by many examples of evolutionary trends which are gradually directed and proceeding along a straight line course. Some of the well-known examples of orthogenesis are the following. 1. The steady increase in the length of horn in rhinoceros-like animal. 2. Fossil line, leading from Hyracotherium, or Eohippus, to Equus, showing trends in changes in a. Increase in body size, b. Reduction of lateral digits, and c. Specialization of teeth. Recently, however, many divergent lineages have been suggested for evolution of horses, see Volpe, 1985. 3. Paludina, snail, fossils showing graded changes of shape of shell. 4. Megaloceros, fossil Irish elk, which had gigantic antlers. 5. Human line of evolution showing 3 straight trends, viz., the increase in size of cranium and brain and the perfection of bipedal locomotion, see Nair, 1985. The selection for largeness, increase in size of body, must have been produced due to the advantage of becoming stronger and resisting predators, e.g., elephants, ungulates and dinosaurs. Some of the progressive series that are observed are explainable as the result of differential growth rates, allometry. Other progressive series are explicable as the result of the operation of natural selection on organisms living in a stable environment or an environment that is changing with a constant trend, e.g., becoming increasingly dry. Under such conditions, natural selection promotes more and more perfect adaptation to that environment, and the resulting changes may take the form of a progressive series. Natural selection operating in this manner is sometimes called orthoselection. Simpson, 1953. Nair, 1985, has differentiated the two terms, orthogenesis and orthoselection, as follows. Thus, according to orthoselection, the change is dependent upon the constancy of the favoring environmental opportunity. However, orthogenesis may involve straight-line progress, which may even go beyond even after the selection has passed its useful stage. RUAVNI, AR018, SNBK. Ecological isolation. A dramatic example is provided by many species of fig wasps, which breeds in, and pollinates, the fruits of a particular species of fig, and each fig species hosts one and only species by the pollinating wasp. Isolation. Solation is an important factor contributing to the process of evolution. It is the segregation of the population of a particular species into smaller units which prevent interbreeding between them. Isolation aids in splitting of the species into separate groups. In consequence of being separated from one another, organisms have developed different characteristics, due to which they have become separate species. Hence, isolation is essential for the formation of new species, i.e., speciation. In fact, besides the variation in heredity, segregation or isolation forms an essential basic factor for the evolutionary process. Isolation is the physical or biotic barrier which prohibits accidental interbreeding. Forms with similar variational tendencies should interbreed to perpetuate them, and dissimilar forms should not, otherwise the new variations would be swamped and unless they were of dominant character would instantly disappear. Thus, gene flow, the sharing of genes between populations, might be suppressed by a physical or geographical barrier. This process is called allopatric speciation. Alternatively, in principle, gene flow can be suppressed in populations which exist in the same area, if, for example, the bearers of favorable mutations mate preferentially with each other and if the selection of these mutations is sufficiently intense to overcome any gene flow from matings with more parental forms. This is sympatric speciation. Finally, even if there is some gene flow between partially isolated populations in adjacent areas, there 114 EVOLUTION, evolutionary biology can, again in principle, be a divergence of characters under the influence of selection if gene flow is relatively low and the differences in selection pressure are relatively high. This is known as parapatric speciation and is likely to be common in organisms with low vagility, dispersal powers, such as plants and land snails, see Callow, 1985. Whereas few biologists doubt the importance of the allopatric process in evolution, there is much debate on the importance of the sympatric and parapatric processes. Isolation is accomplished by two means: physical, e.g. geographical isolation, or biotic, e.g. reproductive isolation, isolations. Geographical isolation is one aspect of species multiplication which Darwin and Wallace failed to understand completely. Its theoretical development starts with the early studies of Moritz Wagner, 1868, 1889, H. C. Bohm, K. Jordan, 1905, and E. B. Pelton, and passes through the subsequent work of Wrench, 1929, Dobzhansky, 1937, 1953, Huxley, 1942, 1954, Stebbins, 1950, and Ford to its culmination by Meyer, 1942, 1949, 1963, 1971. Types of isolation. Evolutionary biologists have recognized the following types of isolations which cause speciation. 1. Isolation by time. The paleontological history of animals and plants suggests that the population at one time is always the descendant of the one living earlier. In case of straight evolution, one species simply gets transferred gradually into a new species through accumulating genetic differences. If the changes are great enough, then the biologists call the population at two different times as two different species. This may well be illustrated with the sequential fossil records of the evolution of horse family. According to G. G. Simpson, horse evolved some 16 million years ago in Eocene. It underwent successive changes produced by thousands of favorable mutations of every gene involved in the evolutionary trend of each characteristic. 2. Isolation by distance, spatial isolation. Sheer distance apart may also act as an isolating factor for a species which occupies a great range of area, which is unbroken by effective barriers. Its example is wrens, birds of South America. Wrens are found all over the continent, but the wrens of one region differ from those of the other in color patterns, size, proportions, and habits. It shows that without any barrier, sheer distance apart tends to produce local races, subspecies. 3. Geographical isolation. It is the most common type of isolation and occurs when an original population is divided into two or more groups by geographical barriers such as river, desert, glacier, mountain, or ocean, all of which prevent interbreeding between them. Then in the course of time, different mutations may become incorporated in the gene pools of the different groups. Often these differences are of such a nature that the separated groups, when they come in contact again, do not interbreed. Thus, species have been formed by geographical isolation. Certain additional geographical isolation. Kinds of geographical barriers. The Kaibab squirrel lives only on the north rim of the Grand Canyon, and B. The Abert squirrel lives exclusively on the south rim. The two populations are geographically separated but still quite similar. Are the volcanic formation of a Mountain on land, mountain. Ranges with deep valleys between and land masses as islands in sea. Isolation 115. The classic example of continuing evolution by geographical isolation is that described by Darwin for finches. Darwin found that there were 26 groups of finches among the Galapagos Islands, which lie a few hundred miles west of South America. Only 5 of these groups were the same as the finches found on the mainland. The other 21 were types peculiar to the groups of islands. Some of the 21 groups interbred quite freely, while others did not. Apparently, each of the groups became isolated by migration. Each group evolved separately from the continental forms as well as from other isolated groups on the islands, forming a series of species and subspecies. Another interesting IRT example of geographical iso. L.W. Laschen is provided by L. F. ant seals. The southern elephant seal, Mirounga leonina, occurs in the cool waters of the southern hemisphere around Antarctica, the southern coasts of South America, South Africa, Australia, New Zealand, and many of the Antiboreal Islands. A close ally, the northern elephant seal, Mirounga angustirostris, is found in cool waters along the coasts of western North America. The two forms are very similar to each other and can be distinguished only with difficulty. However, the breeding populations of the two U.F. pipe. The saola, unknown to science until 1992, is one of a number of previously undiscovered species recently found in the mountains of Vietnam. The area's distinctive assemblage of species probably arose during a past period of forms or separa by Taebo Diuk geographic isolation. 3,000 miles of warm tropical seas, and, hence, are not capable of genetic exchange. Where forms occupy discrete geographic or ecologic ranges separated from one another by spatial barriers, they are called allopatric populations. The two kinds of elephant seals are allopatric species genetically isolated by an eco-geographic barrier. Other examples of geographical isolation can be observed anywhere. It is well established, for example, that there are more different species of the same genus in mountainous country than in plain regions. For instance, in the eastern part of the USA, there are 8 species of cottontail rabbits, whereas in the mountainous regions of the west of the USA, there are 23 species of the rabbits. Often, in mountainous country many of the plants and animals found in deep valleys, which are separated by high peaks but which may be only a few miles apart, are peculiar to those valleys. Likewise, the Isthmus of Panama provides another striking example of geographical isolation. On either side of the isthmus the phyla and classes of marine invertebrates are made up of different but closely related species. For some 16 million years during the Tertiary period, there was no connection between North and South America, and marine animals could migrate freely between what is now the Gulf of Mexico and the Pacific Ocean. When the Isthmus of Panama re-emerged, the closely related groups of animals were isolated and the differences between the fauna in the two regions represent the subsequent accumulation of hereditary differences. Over the long haul, geographic isolation is seldom permanent. Changes in geography, migrations resulting from great population pressure, or chance dispersal during storms may bring separated groups. 116 EVOLUTION Evolutionary biology. In contact again. If they then interbreed and have fertile offspring, speciation has not occurred, but if they do not interbreed or if they interbreed and have sterile offsprings, new species have been produced, reproductive isolation has occurred. 4. Reproductive isolation. In sexually reproducing organisms, species can be defined as Mendelian populations between which the gene exchange is prevented by reproductive isolation. An isolating mechanism is any genetically conditioned barrier to gene exchange between populations. Dobzhansky, 1976. The term isolating mechanism was coined by Dobzhansky in 1937. In simple terms, isolating mechanisms are those which prevent successful reproduction between members of two or more populations, viz., closely related species that have descended from the same original population. Types of isolation mechanisms. Most modern evolutionists such as Mecham, 1961, Meyer, 1948, 1970, Stebbins, 1966, 1971, etc., have classified the reproductive isolating mechanisms into two classes, namely premating or prezygotic isolating mechanisms and postmating or postzygotic isolating mechanisms. Both types of isolating mechanisms differ fundamentally from each other in the following manner. Premating or prezygotic isolating mechanisms prevent wastage of gametes, germ cells, and so are highly susceptible to improvement by natural selection. Postmating or postzygotic isolating mechanisms do not prevent wastage of gametes and their improvement by natural selection is indirect. Meyer, 1970. Both types of isolating mechanisms included many subtypes, all of which have been listed in Table 9-1. Types of isolating mechanisms that prevent gene exchange between population of related species of organisms. Source: Stebbins 1976, Meyer 1971. Table 9-1. A. Premating or prezygotic isolating mechanisms, mechanisms that prevent interspecific crosses, i.e., fertilization and zygote formation. 1. Habitat isolation. The populations live in the same regions but occupy different habitats, so that potential mates do not meet. 2. Seasonal or temporal isolation. The populations exist in the same regions but are sexually mature at different times, so that potential mates remain unable to mate. 3. Ethological isolation. The populations are isolated by different and incompatible behavior, in animals only before mating, so that potential mates meet but do not mate. 4. Mechanical isolation. cross-fertilization or pollination is prevented or restricted by differences in structure of reproductive organs, genitalia in animals, flowers in plants, so that copulation is attempted but no transfer of sperm takes place. B. Postmating or postzygotic isolating mechanisms. Fertilization takes place. Hybrid zygotes are formed, but these are inviable, or give rise to weaker sterile hybrids. 1. Gametic mortality. Sperm transfer takes place but egg is not fertilized. 2. Zygotic mortality. Egg is fertilized but zygote dies. 3. Hybrid inviability or weakness. Zygote produces an F, hybrid of reduced viability. 4. Developmental hybrid sterility. Hybrids are sterile because gonads develop abnormally, or meiosis breaks down before it is completed. 5. Segregational hybrid sterility. Hybrids are sterile because of abnormal segregation to the gametes of whole chromosomes, chromosome segments, or combination of genes. 6. F. Breakdown. F. Hybrids are normal, vigorous and fertile, but F. contains many weaker sterile individuals. Isolation 117. A. Prematting or prezygotic isoiating mechanisms. Prematting or prezygotic isolating mechanisms are those that prevent contact between the species when they are reproductively active, or which prevent or restrict the union of gametes after mating or cross-pollination has occurred. It may be caused by the following reasons. 1. Habitat isolation, ecological isolation. Often two closely related species will thrive in different ecological conditions, habitats, within the same territory, but no hybrid between them will be found. However, when the members of the representative species are taken into the laboratory, hybrids between them can be obtained. Thus, their respective gene pools are isolated physically, but not physiologically. Again, because of the ecological separation that inhibits the sharing of traits, the incorporation of new adaptations may lead to functional separation of their gene pools, and speciation. Habitat or ecological isolation is most common in plants because of their sedentary nature. In them it operates in two ways. The species may live in the same general area but have such different habitat preferences that their populations are rarely close enough together to cross-fertilize each other frequently. In addition,